Now we're going to talk about some of the specific metabolic pathways. And we're going to start with a pathway called glycolysis. And this is the pathway that is responsible for extracting energy from those carbohydrates that we consume in our diet, specifically glucose. Although other carbohydrates or monosaccharides can also and do also feed into the glycolytic pathway, they just come in at a point further down from where glucose starts. Basically, this pathway converts glucose to pyruvate, and then that pyruvate is metabolized to acetyl-CoA. And that acetyl-CoA molecule has several different fates, but one of them is entry into the citric acid cycle. So lipids are metabolized in a different way. Fatty acids are converted to thioesters. We've seen that thioester bond where acetyl or coenzyme A is linked to a fatty acid here, and it undergoes its own metabolic process in which it releases, depending upon the size of the fatty acid, a certain number of units of acetyl-CoA. So we start with some even-numbered fatty acid, and it is cleaved into two carbon unit acetyl-CoA molecules. Amino acids are usually reassembled into new proteins. We do not have a storage molecule in the body for excess amino acids. So we can, however, catabolize them for energy or use them in new proteins. In their catabolism of amino acids, the amino groups are converted to urea, which is then excreted in urine. So we don't want a buildup of nitrogen in our tissues. So we've talked about enzymes, and we've discussed the fact that the name of the enzyme often gives us an idea as to what that enzyme does or the reaction it catalyzes. Excuse me. So carboxylase catalyzes the addition of a carboxylate ion. Carboxyl, okay, carboxylate. And that ASE tells me it's an enzyme. So carboxylase, decarboxylase, this enzyme removes a molecule of CO2. Dehydrogenase, just the way it sounds, hydrogen dehydrogenase. We remove 2 atoms of hydrogen. Isomerase, we know what isomers are, so this enzyme catalyzes the isomerization of one isomer into another. Finally, a kinase. This catalyzes the transfer of a phosphate group, and we'll see that in what we call substrate level phosphorylation of ADP to form ATP, and we'll also see kinase activity in phosphorylating substrates or intermediates in a number of pathways. The transfer of a phosphate unit from ATP to fructose 6-phosphate is catalyzed by a kinase enzyme. So fructose 6-phosphate is an intermediate in the glycolytic pathway, and the enzyme that catalyzes the transfer of a phosphate group from ATP to the 1 position in fructose 6-phosphate. So here's the number 6 carbon with its phosphate group. the enzyme is called phosphofructose. So it's telling us that kinase is adding a phosphate group to a fructose molecule. And here we are with the product, fructose 1,6-bisphosphate. So kinase transfers a phosphate group. So glycolysis is a linear 10-step anaerobic pathway responsible for converting glucose, a 6-carbon saccharide, monosaccharide, into 2 3-carbon molecules of pyruvate. If you remember, we looked at 2 different possible pathways for these pathways, metabolic pathways. One could be linear. We looked at linear, but the other was cyclic, and we're going to see that in the citric acid cycle. We can break down glycolysis into 2 phases. Okay. There is the energy investment phase, which comprises steps 1 through 5. So we have to utilize 2 ATP molecules. So we have phosphates hydrolyzed from those ATPs, and ADPs are released. And then the 6-carbon glucose is converted into 2 3-carbon segments. And during the second phase, we start generating energy. Okay, so for each pyruvate that's formed, of which there are 2, we generate 2 ATPs and 1 NADH. Remember, this is the reduced form of NAD+. So the energy investment phase uses 2 ATP, and the energy generating phase produces a net, well, not a net, a total, let's say, of 4 ATPs. However, we use 2 here, so we net 2 ATPs for the catabolism of 1 glucose molecule. So this is kind of an overview of glycolysis, the first phase, or the energy investment phase, showing where we are hydrolyzing our ATP to phosphorylate an intermediate in that glycolytic pathway. And the products, the initial 3-carbon products, are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This DHAP will get converted to glyceraldehyde 3-phosphate, and then they will both be treated the same way in the rest of the pathway, or the second phase. So here's that second phase. This glyceraldehyde dihydroxyacetone, or DHAP, is converted to glyceraldehyde. So now we have 2 of those, and they undergo the same steps until they are converted to pyruvate. And you see we produce NADH and 2 ATP for each of the individual glyceraldehyde 3-phosphates. So you're not responsible for the individual reactions in glycolysis, but you are responsible for understanding the kinase or the enzyme activities and how that contributes to the extraction of energy. And step 1 begins with the phosphorylation of glucose on carbon 6. So we have added a phosphate group from ATP, and hexokinase is the enzyme responsible for that. It is a kinase that phosphorylates a 6-carbon carbohydrate. And the purpose of this phosphorylation is that it actually gives glucose a negative charge, or glucose 6-phosphate a negative charge. And that keeps the glucose in the cell, so it can't escape. Second step, there's an isomerization. So we're just rearranging glucose 6-phosphate to fructose 6-phosphate. Okay, so we've got an isomerase as our enzyme, phosphohexose isomerase. So we have a 6-carbon carbohydrate that has a phospho group on it. Then we have another phosphorylation. So we have phosphofructokinase. We saw this reaction a minute ago, where we phosphorylate carbon 1 in the fructose 6-phosphate molecule. So the 1 and the 6 then again are identifying what carbons those phosphate groups are on. And the first 3 steps are to add the 2 phosphate groups, isomerize a 6-membered ring, glucose, to a 5-membered fructose ring. So remember, with that isomerization, we limit that ring to 5 carbons. And we produce energy in the form of 2 ATP molecules, but we have utilized, energy stored in the 2 ATP molecules. We're using energy right now, is used to modify that glucose. But that contributes and helps the generation of energy in later steps. Step 4, we're going to cleave, or we're going to cut that fructose 2,6-bisphosphate into 2 3-carbon molecules, and an aldolase, or aldolase is the enzyme responsible for this cleavage. So this ring has to open up, and then we, whoops, sorry about that, didn't mean to flip that. We cut that into the 2 3-carbon molecules, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. We isomerize DHAP with the help of triose phosphate isomerase. So this is an isomerase that works on a 3-carbon molecule that has a phosphate group. So the first phase of glycolysis then converts glucose to 2 glyceraldehyde 3-phosphate. In step 6, we're going to oxidize and phosphorylate that glyceraldehyde 3-phosphate. So now we have 1,3-bis phosphoglycerate, and here's the oxidation reduction. This is reduced. This is again the oxidized form, NAD+, so it's reduced. It picks up a hydrogen and 2 electrons, and we add a phosphate group. One 3-bis phosphoglycerate then contributes a phosphate group to ADP. So now we're going to start regenerating ATP. So phosphoglycerate kinase catalyzes what's called a substrate level phosphorylation, and one of these phosphate groups is transferred to ADP to produce ATP. And we are left with 3-phosphoglycerate. 3-phosphoglycerate undergoes an isomerization, and basically the phosphate group is moved, and we call this enzyme a mutase. So I think in general they call it an isomerization, but I like to think of it as an enzyme that allows or contributes, catalyzes the migration of some substituent group to a different place in the molecule. And that's what happens here. We convert 3-phosphoglycerate to 2-phosphoglycerate. Step 9 is a dehydration reaction, and this is catalyzed by the enzyme called enolase. So 2-phosphoglycerate is dehydrated to phosphoenolpyruvate, or PEP. This is a very high energy molecule, and it will be converted to pyruvate in the last reaction, or the 10th reaction. And what happened here in this dehydration was we removed a molecule of water and created a double bond between 2 carbons. The last reaction in glycolysis, a phosphate again is used or transferred by pyruvate kinase to ADP to produce that ATP. So this is another substrate level phosphorylation, and because we started with 2 glyceraldehyde 3-phosphates, we end up with 2 pyruvate molecules. So the second phase essentially summarized: 2 glyceraldehyde 3-phosphates are converted to 2 pyruvates, and from those 2 glyceraldehyde 3-phosphates we form 2 NADHs and 4 ATPs. The net result, of course, is we use 2 ATPs in phase 1, we produced 4 in phase 2, so the net gain of ATP from glycolysis is 2, and 2 NADHs. These NADHs are going to be transported to the mitochondria and will enter the electron transport chain, I can't talk today, and make more ATP. The fate of the 2 pyruvate molecules depends upon whether or not O2 is available. And if it is available, that pyruvate will enter the citric acid cycle. If it is not, it will be converted to lactate. And this is just the chemical equation for the net result of glycolysis, where 1 glucose molecule plus 2 NAD plus produces a net of 2 ATP, pyruvate, 2 pyruvates, 2 NADs or NADHs, and 2 protons. And that is the glycolytic pathway.